So yes, hi, I'm Matine Rubin. My project is on the omics analyses of the effects of combustible and electronic cigarette smoke. I've done research under Dr. Prue Talbot as my faculty mentor in the Department of Molecular Cell and Systems Biology. So for some background, cigarette smoking has a high potential to cause severe diseases, and they're hard to quit due to their high addiction value due to the high nicotine content. So e-cigarettes are a cig-alike. They mimic the effects of cigarette smoking, and the use of e-cigarettes is associated with smoking cessation in adults who did not plan to intend to quit smoking. And although commonly believed to be less harmful than regular cigarettes, the liquid aerosolization process heats the e-cigarette liquid to such high temperatures that they generate toxic substances within the device that is later inhaled by the user. So our first experimental question here is, would continued e-cigarette use after smoking cessation lead to the reversal of harmful effects caused by the previous cigarette smoking? And our second experimental question here is, how would dermal exposure to nicotine affect the skin? So in addition to inhalation, nicotine uptake can occur via dermal exposure as well. This could be as a result of e-cigarette malfunction, which would cause the fluid to leak out of the device onto the hands of the user or other surfaces which are then touched. So little is known about the effects of this dermal nicotine exposure caused by e-cigarette liquid. So our second experimental question here is, how would the protein expression of nicotine-treated 3D model of human skin be altered? So again, a recap of our research purposes here. So our first aim is to investigate whether converting to e-cigarettes for at least 6 months was a healthier alternative to continued combustible cigarette use. And we're analyzing this through gene expression analysis with 3 different experimental groups. So first, it would be people who are former smokers, but now using e-cigarettes. Second would be current cigarette smokers. And then our third experimental group is non-smokers. Then our second experimental aim here is to understand how dermal exposure to nicotine affected protein expression. And this again was with the 3D human skin model called Epiderm, and we tested low and high dosage concentrations of nicotine. For the sake of time though, I'll just be focusing on our first experimental question here. So the in vivo experiment will be omitted from this presentation. So to get into the demographics of the participants of our e-cigarette analysis, so 22 nasal epithelium biopsies were collected from e-cigarette users who are former smokers, current smokers, and non-smokers. 19 of those samples were chosen to undergo the RNA sequencing analysis, and then after data cleaning, each group contained data from 3 participants. So here we can see that in the table, 3 e-cig users who were former smokers, 3 non-smokers, and 3 cigarette smokers. And on the far right here, you can see the urine cotinine concentration that was used to verify the status or the reported status of these participants. So we used a cutoff value of 400 nanograms per milliliter of cotinine, and cotinine is a metabolite of nicotine. So if you have a high concentration of cotinine in your urine, it indicates that you've been exposed to nicotine. So here we can see that the e-cigarette users all had above 400 concentration of cotinine in their urine, as well as in the cigarette group, and then the non-smokers had 0 or non-existent readings for the urine cotinine. And we also have here the puffs per day of the e-cigarette users and the cigarettes per day of the cigarette users. And our further methods to analyze the data that was collected from the nasal epithelium biopsies, we used the software called Ingenuity Pathway Analysis, IPA, by QIAGEN, and this is powered by QIAGEN's Ingenuity knowledge base to identify significant diseases and biological functions, canonical pathways, and regulator effects that were observed as a result of the experimental treatment. And prior to analyzing our data with IPA, we fed it through this lung filter. So we filtered our data to only include genes that were related to lung function and lung health to make our research more focused and get more focused results from IPA. And then another software we used was DOSE, which is Disease and Ontology, Semantic and Enrichment. This is a package in R. And with this, we used the package to identify associations between differentially expressed genes or DEGs and disease ontology terms. So first we can look at the results that IPA produced. So this table shows the top biological functions for electronic cigarette users versus non-smokers. So again, these are former smokers who are currently using e-cigarettes. And this is lung filtered data again. And the top 9 biological processes that were upregulated, so that means that there was a positive Z-score, indicating that the e-cigarette group had increased mostly inflammatory and immune reaction. So besides this lung cancer disease that was found, every pathway that was upregulated besides that was related to immune and inflammatory response. And then the one pathway that was decreased in this comparison group was related to formation of cilia. So that table was produced by IPA. These CNET plots were produced by DOSE and R. And this is, here we're looking at a CNET plot of upregulated DEGs in the SIM comparison group. So this is electronic cigarette users versus non-smokers. So here, this is the upregulated one. So this also indicates upregulated pathways here and upregulated diseases. So first, this agrees with our IPA produced data that there would be an increased inflammatory and immune response with, it's printed here, but it's kind of small, so I rewrote it here. So this includes cell surface interactions with the vascular wall, IL-4 and IL-13 signaling, and neutrophil degranulation. And this CNET plot here, so the disease ontology terms here are represented by these kind of beige nodes, and the size of those nodes depends on how many genes are associated with the disease. So the genes that were found in our data to be upregulated and related to these diseases fill in the rest of the circle here. And then you can see with the different color coding, you see which disease they're associated with. And in the CNET plots, we can really get a handle on how many diseases are overlapping into the many, or how many genes are overlapping into their association with the different diseases. So this was a way we visualized data from DOSE. And continuing with the upregulated DEGs here and the upregulated diseases, the DOSE analysis produced results that indicated that there was an increased keratinization response as a result of e-cigarette use. And the related diseases here were formation of the cornified envelope and keratinization. So this was not seen in IPA, but still found to be significant in the DOSE analysis. And here we are looking at the downregulated analysis in DOSE. So these were the downregulated DEGs in the same comparison group with e-cigarette users versus non-smokers. So all of the significantly identified disease ontology terms were related to cilia, and this supports our conclusions or our results found by IPA. So again, here we're looking at the disease terms in beige, these nodes here, and then the genes that are connected and mapped onto those disease nodes. So while we're looking at the effects of chronic e-cigarette inhalation, we're looking at the human transcriptome. And in previous data has shown that individuals who had ceased smoking combustible cigarettes for 14 days exhibited decreased inflammatory response comparable to non-smoker levels. However, our data show that there was a continued inflammatory and immune response. So we speculate that continued e-cigarette use after smoking cessation was likely the cause of this prolonged inflammation that we observed. And we also can conclude that e-cigarette use for 6 months did not reverse the effects of inflammatory effects of cigarette smoking. And this is an issue because smoking-related inflammation has correlated with severe lung damage. And we also found previously that increased keratinization leads to squamous cell metaplasia in response to cigarette smoke, and this may lead to cancers like bronchial carcinoma. And this is squamous cell metaplasia is characterized by the thickening of epithelium And this continues during e-cigarette use, which is seen in our data due to the increased keratinization that we found in the results from dose. And this further indicates that 6 months of using e-cigarettes was not able to reverse the effects of keratinization on epithelium in the lungs. Lastly, e-cigarette inhalation is detrimental, cigarette smoke inhalation is detrimental to cilia health and formation of cilia. That was found by a previous researcher. And this is an issue because lack of cilia in lungs and airways impairs the ability of the lungs to remove bacteria, pollutants, and mucus, which could lead to infection. And again, our data showed a decrease in cilia function and formation of cilia. So we could conclude that 6 months of e-cigarette use did not reverse the effects that cigarette smoking had on cilia health. And these effects continued with e-cigarette use. So in conclusion, again, we looked at the nasal epithelium biopsies of 3 former smokers who are now currently using e-cigarettes for at least 6 months. And we saw an increased inflammatory response, increased keratinization, and decreased formation of cilia and cilia health in general. So we can conclude from these results that switching to e-cigarettes completely from combustible cigarettes for at least 6 months did not reverse the effects of previous smoking. And further investigation is obviously necessary to better understand the effects of e-cigarettes, as well as to inform public health measures related to these devices in the future. And results from, or the data from this experiment, as well as the dermal exposure experiment that I was unable to talk about here, are written into 2 publications that are in review now and on their way to being published. So I included those references there. And here are my references I utilized in the presentation. And then lastly, I wanted to thank Dr. Talbot, Dr. Gio Pozuelos, and the graduate students from Dr. Talbot's laboratory, in addition to their collaborators, for conducting the primary research that produced the data I used for these investigations. And I also wanted to especially thank Dr. Talbot for her continued mentorship throughout this process. Lastly, I wanted to thank University Honors for providing funding for this research. And thank you so much for listening, everybody.